Hello. Today, I'm happy to welcome Sébastien Depienne for this new episode of my publication at a glance. Sébastien performed his PhD at the University of Nantes, working in the Sésame laboratory, and he's going to present his last article published recently.
Thank you for the opportunity. So our recent publication is basically about the functionalization of the proteins that are present at the surface of viruses, bacteria, and cells. And to achieve this, we used an electrochemical bioconjugation reaction that we recently developed. So first, it actually is interesting to functionalize their surface proteins because they are involved in many biological events, and the deeper understanding of those relies in our ability to probe or to control their interaction framework. On the other hand, the engineering of viruses, bacteria, and cells is extensively studied for gene, bacterial, and cell therapies, and the surface labeling of those biosystems is then a way to improve their efficiency by giving them new properties such as immunogenicity regulation or tissue targeting to prevent the side effects, for instance. And in this context, the functionalization strategies currently heavily rely on genetic manipulations that are efficient but associated with expensive and complex productions, or on metabolic engineering, which is based on several days of incubation. The direct bioconjugation of the surface proteins has been very poorly explored, mostly because of the difficulty to put these biosystems in presence of chemicals or conditions that are potentially harmful. So considering all of this, we recently developed a fully biocompatible electrochemical methodology to label peptides, enzymes, and clinically relevant antibodies. The proof of concept and the development studies are available in those two publications from the group, and I will now talk about how we could extend this methodology for the fast and simple direct surface bioconjugation of viruses, bacteria, and cells. So the principle is to have an aqueous solution of the biosystem of interest, and this solution also contains a non-reactive molecule, which is here the N-methylluminol, and which has a low oxidation potential. So by driving a simple 3-electrode system inside the solution, a controlled weak electrical current is applied thanks to a small potentiostat, and this results in the selective electro-oxidation of the N-methylluminol without affecting the biosystems. The activated species that is generated progressively is then able to react directly with the surface proteins and more precisely with the tyrosine residues. So we explored the electrochemical functionalization of the adeno-associated virus of serotype 2, the bacterial strains Escherichia coli and Staphylococcus epidermidis to study both gram-negative and gram-positive strains, surfaces, and two cell lines, HEK and LR. For the virus, we choose to label it with the GalNAc and mannose carbohydrates because they are relevant to target selectively the cells in the liver and in the retina. And for bacteria and cells, we demonstrated the functionalization with the interesting here acido bioorthogonal label, which could be further fluorescence detected using strain-promoted click chemistry in both cases. So different types of electroactivations were investigated, and satisfyingly, the covalent conjugation could be confirmed for all the biosystem studied with 4 different N-methylluminol derivatives, including the biotin luminol, and so in a reproducible time-controlled manner for all cases, and with reactions as short as a few seconds or a few minutes for these conditions. We also could visualize the clear surface labeling of bacteria and for the cells using fluorescence and confocal microscopies, giving those exciting images. And afterwards, we evaluated the integrity of the electro-engineered bioorganisms, which is obviously of most importance. On the one hand, we were pleased to see that the assembled capsid of the viruses was preserved in all cases, and that the ones that were not too much functionalized were still able to transduce efficiently the cells. And on the other hand, in culture conditions, both electrolabeled bacteria and cells completely conserved their ability to divide, meaning that they were still fully viable. So considering this general efficiency, the kinetics observed, and the bioviability, we believe that the click electrochemistry tool has the potential to be very beneficial to investigate surface biological events, as well as to develop the next generation conjugates for gene, bacteria, and cell therapies. So this is where this study ends. Don't hesitate to check the publication directly for more details, and thank you for listening.